everyone, today we are going to see the topic glucose metabolism. So there are two main sources of glucose. One is production and another one is utilization. So in production the three process takes place, that is the three cycles takes place: glycogenolysis, gluconeogenesis and exose interconversion. Where in utilization it undergoes glycogenesis, glycolysis, which then leads to Krebs cycle, hexose interconversion and HMP pathway. So coming to the metabolic pathways, there are two cycles: catabolic cycles and anabolic cycles. So the glycolysis, Krebs cycles, HMP, glycogenolysis comes under the catabolic cycles, where glycogenesis, gluconeogenesis comes under the anabolic cycles. So glycogenesis is the synthesis of glycogen from glucose. It is mainly takes place in liver, muscles and cytosol, where glycogenolysis is the degradation of glycogen into glucose, which also mainly takes place in liver, muscle and cytosol. So the main major objectives of glycolysis, it is considered as major oxidative pathway of glucose. So the main reactions of the glycolytic pathway and the ATP production has happened in both the aerobic and anaerobic, and pyruvate kinase deficiency which causes hemolytic anemia. So glycolysis, the major pathway for glucose metabolism, mainly occurs in the cytosol of all the cells. So it is unique and it is function as both aerobically and anaerobically depending on the availability of oxygen. So it allows tissues to survive in the presence or absence of oxygen, and RBCs which lack mitochondria is completely reliant on glucose as their metabolic fuel and metabolize it by anaerobic glycolysis. So these are the overall structure, where glucose 6-phosphate is obtained from the glucose and glucose 6-phosphate converted into fructose 6-phosphate, and fructose 6-phosphate converted into fructose 1,6-bisphosphate. There occurs two major divisions: glyceraldehyde 3-phosphate and dihydroxyacetone. So from glyceraldehyde 3-phosphate 1,3-bisphosphoglycerate, and from 1,3-bisphosphoglycerate converted into 3-phosphoglycerate, then 2-phosphoglycerate and phosphoenol pyruvate, and finally pyruvate that converts into lactate. So coming to the steps, brief we'll see. So first, glucose is converted into glucose 6-phosphate, the action of enzyme hexokinase, where ATP is converted into ADP. And the next step, with the help of phosphoglucose isomerase, glucose 6-phosphate is converted into fructose 6-phosphate, and the fructose 6-phosphate with the help of enzyme phosphofructokinase, which converted into fructose 1,6-bisphosphate by the action of aldolase enzyme. And aldolase enzyme, there are two subdivisions are there: glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So glyceraldehyde 3-phosphate is converted into dihydroxyacetone phosphate through the enzyme triose phosphate isomerase, and the glyceraldehyde 3-phosphate with the help of enzyme glyceraldehyde 3-phosphate dehydrogenase converted into 1,3-bisphosphoglycerate, and the phosphoglycerate kinase enzyme converted into 3-phosphoglycerate from 1,3-bisphosphoglycerate, where 3-phosphoglycerate is converted into 2-phosphoglycerate by phosphoglycerate mutase, and 2-phosphoglycerate converted into phosphoenol pyruvate with the help of enzyme enolase, and phosphoenol pyruvate with the help of pyruvate kinase, it gives a product of pyruvate. So coming to the glycolysis, the end product is pyruvate That is 2 molecules of pyruvate and 2 molecules of ATP and 2 molecules of NADH. Thank you.